So I want to start by just clarifying the basic question, what is the human genome? Well, the human genome is a long DNA biopolymer, but it's, if you believe it or not, it was only in 2003 that we were able to figure out the draft reference sequence of the human genome, which is essentially about 3 billion nucleotides or letters in length. So it's 3 billion A's, C's, G's, and T's. Okay? And that was a seminal moment because it's the first time we could see the blueprint of what makes us human. But if you think about it, it took essentially $3 billion and decades of work to get one draft of the human genome. Now, fast forward a few years later, and there's been a dramatic improvement in sequencing technologies that has reduced costs. So today you can sequence a single human genome about $1,000. And hence, there has been a super exponential growth in the number of human genomes. Now, when I talked about the reference human genome that was sequenced in 2003, it was sort of an average genome across all individuals. Now, we are not all identical. We all have differences. And actually, the way we see this is if we sequence thousands and thousands of individuals, what's referred to as population sequencing, then we start seeing positions in the genome where we differ. These are called genetic variants. Okay? And there's lots of genetic variation. About 3 million positions in the genome vary across individuals. Most of them are probably benign, but a handful of them are not. And so using interesting case control studies, where you sequence patients and you sequence controls, you can now start identifying genetic variants, mutations, that are significantly associated with diseases. This has been very popular. It's been done for thousands of diseases. And we have a good collection of thousands of genetic variants that we believe are associated with disease, show statistically significant association. But let me argue that sequencing is not enough, because essentially what you get when you sequence an individual is what you see on that screen, a bunch of letters. And we need to really understand what each of these letters means. So what does, for example, this particular A do in the genome sequence? And what does that particular word do in the genome? So that's where we come in. Our goal is to really take genome sequences and understand what they do. What does every letter, every word in the human genome actually do, or does not do? The reason this becomes even more interesting is because humans are multicellular organisms, right? We have thousands of different cell types and tissues in the human body. But really interestingly, every single cell type and tissue has the exact identical genome. So how is this single sequence encoding this kind of diversity of function, right? And actually the answer is quite simple. For that, we need to figure out what's happening inside the genome. So let's look at genome anatomy for a moment. So let's take the DNA in our cells and unwrap them. And we want to understand what are the different kinds of functional elements in the genome, little pieces of the DNA that do interesting functions. The most common functional unit in a genome is genes. So genes are essentially pieces of DNA, as shown on the slide, that encode for proteins. Okay? So these genes are read and they produce specific proteins. There are only 20,000 genes in the human genome. That's it. Okay? And they produce a variety of different proteins. And they form the ingredients that can create whichever cell type you want. So you mix and match different genes, right, which produce different proteins, and you get different cells, cell types. Okay? But these are the ingredients, and to produce or not produce specific collections of genes, you need a complex control circuitry, right? This control circuitry is also encoded in the genome. So, as shown in the slide, the little elements that you see, the yellow and the red elements, these are control elements that control precisely which genes produce which proteins in which cell types. So they define cell type identity. Now we've known where the genes exist for a long time and what they largely do, but the control elements have been largely elusive. Now using sequencing technology, we've been able to do various kinds of biochemical profiling experiments that can allow us to very precisely identify where different kinds of biochemical events are happening in the genome. So for example, in this slide, you see a black triangular protein. It binds specific positions in the genome. We can do an experiment that gives us a genome-wide track at every nucleotide, every position in the genome. It gives us a score of the likelihood of observing that particular protein at that position. And we can similarly do multiple experiments with different kinds of biochemical markers. Now you'll start seeing some interesting correlations. The gene on the right shows a really nice profile of this green modification, this green biochemical marker, precisely marking the gene boundary. Similarly, the orange and the red markers, they very well define control elements. So using this strategy, the NIH funded 2 large projects, and they allowed us to essentially perform these kinds of profiling experiments, genome-wide profiling experiments, in hundreds of different cell types and tissues. Okay? So what you end up getting is this giant data cube, where one of the axes corresponds to 3 billion genomic positions, the other axis corresponds to hundreds of biochemical measurements, and the third axis corresponds to hundreds of cell types and tissues. So now we are in a data problem, right? We have millions of terascale data points, and we're trying to make sense of them. We're trying to reverse engineer functional units in the genome. That's where we come in. So we apply machine learning and deep learning approaches to identify tissue-specific control elements that I showed you before, identify how genomic sequence encodes these control elements, and then can we use this information once we've decoded the genome to understand how genetic variation associated with disease may be affecting molecular function. So let me give you some interesting findings that we came up with. So as I mentioned to you, the control elements in the genome were largely elusive. Now, after integrating this massive data cube using various sort of standard machine learning techniques like hidden Markov models, we were able to infer that there are essentially 2.3 or so million control elements. So you have only 20,000 genes, but you have 2.3 million control elements controlling those genes. Okay? And those control elements are not active in every cell type. So in the figure I show you in the slide, the columns represent the 2.3 million elements, and the rows represent various cell types. And if you cluster the activity of these elements in the different cell types, you see beautiful modular behavior, right? You see mostly a diagonal pattern. So there are specific collections of control elements that activate in very specific cell types and tissues, giving rise to cell type identity. So now we've sort of decoded which portions of the genome are controlling gene activity, and hence we've discovered sort of new elements, new functional elements in the human genome. But what we're more interested in is how the DNA sequence of these control elements defines their cell type specific activation. Now the way this works is there are specific DNA words, as shown on the slide, to which proteins like to bind. Okay? And by placing specific DNA words in different positions in the genome, you can precisely, it's a code, you can precisely control which proteins bind those positions and activate them in specific cell types and tissues. And this code is largely unknown. So this is where we can essentially use machine learning to figure out how the DNA sequence is encoding this beautiful dynamic patterning of activity of these control elements. So the idea is kind of simple. You take these 2.3 million elements, you look at their differential activity states in different cell types, and then you try to infer specific patterns in the sequence underlying those collections of elements that behave similarly to identify these DNA words that are the functional units encoding function in these control elements. So essentially, you can think of this as a machine learning problem where you have 2 classes of sequences. One class of sequences corresponds to control elements specifically active in tissue number 1, and the other class of sequences correspond to control elements that are active in all the other tissues, right? So you have a nice binary classification task. The goal is to take these sequences and try to predict which class they belong to. This is where we turn to deep neural networks, because essentially we can start off with the raw DNA sequence as the input and try to predict whether that specific sequence is active or not in a specific cell type. So the reason we like these neural nets is because essentially they're able to induce interesting patterns. In this case, we're looking at a specific class of neural networks called convolutional neural networks, and they're able to learn pattern detectors that essentially correspond to DNA words. If you visualize these pattern detectors, they look exactly like probabilistic DNA words. And the lower layers in the network can learn these words, and the higher layers can integrate these words and come up with higher order patterns, what we refer to as genome grammars, combinations of words that define whether that specific sequence is active or not. And by using multitask architectures, we can simultaneously train giant models on all the cell types simultaneously, so that we can take a single sequence and predict whether it's active in each of the hundreds of cell types and tissues. Okay? So we can do this prediction task, and when we actually go and look at what the model learns, we in fact firstly find that we get very good prediction performance, so the model is able to accurately capture this activation information. But more interestingly, when we look inside the model and look at what the pattern detectors learn, we identify hundreds and thousands of interesting DNA words. Okay, so the neural network is able to infer from the raw DNA sequence by looking at these dynamic activity patterns what these interesting DNA words are. We further developed interesting methods that can take any individual sequence, okay, and decompose precisely which nucleotides, which position in the sequence, are contributing to that prediction of that sequence being active in a particular tissue. So the algorithm that we developed is called DeepLift, and my student Avanti will be presenting this in poster number 7 during the poster session. So essentially, you can think of taking every sequence, picking a specific cell type, and predicting precisely in which cell type that particular sequence is active, and then backpropagating this prediction, decomposing it back to individual nucleotides in the sequence, and predicting which pieces of the sequence are actually relevant. So by doing this, so for example, we can take a specific sequence of a control element, and the neural network, once we are able to backpropagate the importance scores at the level of individual letters, you can see essentially in this sort of long sequence, there are 4 words that really pop up. So this is an example of a combinatorial grammar. We have 3 instances of what's called a GATA word, and 1 instance of an SPI 1 word. So it's these 2 proteins that come together and jointly activate this particular sequence. Okay? And we are able to do this for millions of different control elements in hundreds of cell types and tissues. So we're really able to decompose precisely these interesting genomic grammars that control gene activity. More interestingly, we can take genetic variants associated with disease, as shown here. So this is a genetic variant in the genome where a C to a T transition, so if you change the C to a T, essentially that variant seems to be highly associated with coronary artery disease. We want to understand the mechanism through which this is working. Okay? We know where the variant is, but we don't know exactly what it does. Now, if we take the variant and we look at its prediction of the neural network in unstimulated coronary smooth muscle cells, the neural network says, oh, that particular position is not really important. So coronary smooth muscle cells are really important tissue type for understanding coronary heart disease, but the neural net says it's not very relevant. However, now if you switch to stimulated coronary smooth muscle cells, all of a sudden, the neural network now says, oh, there's a really important DNA word right there. It's a binding word corresponding to a very specific protein, and if you mutate the C to a T, you actually see a dramatic decrease. If you see the column to the right at the bottom, you see a negative 13.2 decrease in the probability that that element will activate. Right? So if you started from a single genetic variant, we can predict that it's specifically manifesting itself in stimulated coronary smooth muscle cells through a DNA word corresponding to that TCA, TGA word, which corresponds to the binding site of a specific protein, providing us a potentially interesting drug target going forward. Okay? And so we are doing this for a very large collection of genome-wide association studies, these studies that give us large collections of genomic variants associated with disease. And the reason this is really interesting is because only 5% of genetic variants associated with complex diseases actually break genes. The rest of them are very likely breaking control elements. So 95% of disease-associated variation we can now interpret using these kinds of models. So I'll just stop there and sort of briefly outline sort of what we're planning to do. The future of personalized medicine as we see it, we think essentially you'll have to do large-scale personalized genome sequencing. You'll have to integrate that with personalized functional genomics, these giant data cubes that measure various kinds of biochemical activity, integrate that with clinical and electronic medical health records, and develop interesting domain-specific machine learning interpretable models that allow us to rapidly interpret personal genomes, make personalized diagnosis, help with drug target design and identification, and also optimize treatment regimes. And to enable this particular strategy, we want to bring everyone into the fold, not just computer scientists. There are a lot of biologists who could be extremely useful in terms of making this goal happen. And so we've developed interesting tutorials to try to get folks who are not essentially highly trained in deep learning to start building and improving models for genomics. And that's one of the toolkits we've developed. And my student Johnny will present this in poster number 21. So I recommend you go and check it out. Thanks a lot.
Thanks, Anshul. Questions for Anshul? We have a question right there. We're making you work hard, Trudy.
So I had, I guess, two questions. So the
Yeah.
first is, how are control elements represented in the body, and are they a fixed set? That is, do they vary from person to person?
Great question. So there are actually little pieces in the genome, as I showed you. There are little sequences in the genome that essentially encode these. They have these DNA words that correspond to these binding locations of various proteins. So they're about, let's say, about 100 to 200 base pairs long. That's about the sequence size. And there are millions of them scattered all over the genome, okay? And even though you can think of them as a fixed collection, like I said, about maybe 2 to 3 million of them, they have highly tissue-specific activity, as I showed you. And you're right in the sense that the 3 million genetic variants I talked about that are maybe potentially benign, right? Those can, in fact, affect whether those elements are active or not. Just by disrupting an element doesn't mean you'll necessarily have a disease. There's a lot of robustness in the human genome, right? So it can tolerate a lot of random genetic variation. So there are differences across individuals too. So some elements will be active in one person, not in the other, which will potentially predispose them to disease or not. So hopefully that answers your question.
Other questions?
Yeah, I have a question here. Yeah. So compare on this side. Yeah. So
yes.
compare with the industry like human longevity or Grail. They have a lot of data. They have a lot of resources. Compare with them as a lab. How can you complementary or compete with them?
Great question. So this is where open science comes in. So I talked about two of these consortia, NIH-funded consortia. They've been funding projects for about 2 or 3 years, producing these massive data cubes. So it's a large consortium with thousands of different labs from all over the world, researchers, and it's essentially entirely open data. So there's no embargo. Even before publications come out, the data is released completely openly so that it can be used by anyone. I think that kind of model for data release is extremely powerful, and it even goes beyond what individual companies could do.
Any other questions for Anshul? Great. Thank you very much.